what you have heard also, what you have heard also in last interdrought is that, there are some environments where we can identify that the transpiration efficiency actually partially explains the plant production. But there is a lot of environments where this relation simply doesn't work. This means that the processes driving transpiration efficiency are very likely dependent on the environment of growth. And this is the main topic which I really want to talk about today. But this also has one very important implication, and that is that high transpiration efficiency will simply not work everywhere for improvement of crop production. So that brings us and opens the research questions which we are solving now at ICRISAT. So we have heard a lot about the differences in the plant capacity to channel the water during the changing vapor pressure deficit during the previous talk. So I will not talk about this much further. But what we are gathering a lot of evidence about is that the plants which have the capacity to limit their transpiration upon raising vapor pressure deficit, upon the raising evaporative demand for transpiration, are indeed those which have more likely higher transpiration efficiency and vice versa. On the other hand, we have gathered quite some important evidence that these mechanisms are actually linked to the way how plant is channeling water. And we strongly believe that aquaporins are involved in these processes. And we have heard a lot about it previously, so again, I will not go too much into the details here. But what is important message here? Nowadays, what starts to appear is that all these processes are really dependent on the environment where plant growth and development takes place. Because if we grow the plants in a high VPD, we can see that these plants are not really responding and limiting their transpiration upon rising VPD, while where we grow these plants in a low VPD, then we can see very clear limitation at certain vapor pressure deficit. So this is one part. Yesterday also we have heard a lot about the root structure and tissue structure and how it could affect the channeling water through the plant tissues. So we have gathered some evidence that in this case, root structural and proportional dimensions of the cell roots in the root tissues, in this case, the dimensions of the endodermis, really explain how plant channels its water at higher VPD regimes. And this is not the only evidence. We are also gathering the indirect evidence that we can distinguish the same plant types based on the inhibition of water flow through apoplast. So building upon this, we also observe that these processes and particularly the cell dimensions are different if we grow plants in a different conditions, which means that these processes are also interacting with the environment of growth. So we don't want to stay at the level of the roots only. We were interested whether the similar processes are actually affected at the level of the leaves. So we were playing with the hypothesis that the leaf expansive processes are also affected by the environment of growth. And indeed, you see these genotypes. When we grow them in low VPD, then there is a huge difference in leaf length, which these genotypes attain. But if we grow them in high VPD, there are virtually no differences between the leaf sizes and leaf length in these genotypes. The same is true if we measure the total plant leaf area. So you see that this genotype A, it develops a similar canopy size across very different VPD regimes, but the genotype B has really huge differences or develops the canopy very differently if it is grown in a different VPD conditions. So this means one and single thing, that even the canopy expansive processes are affected by the environment of growth on a really long term. So summarizing on these two first parts, my humble opinion on this is that transpiration efficiency is really the function of the genotypic propensity to channel the water and expand its tissues in the context of given environment. So just to remind you, I was talking about water transport, how it affects the plant transpiration responsiveness to VPD, and that it's very likely linked or one of the underlying processes influencing transpiration efficiency. I was talking that root structure is also likely to influence the plant transpiration response to VPD, and therefore also likely linked to transpiration efficiency. And since the similar processes are affected at the level of the canopy, then it is not that much inconceivable that even the canopy expansion processes has the role to play in the final values of transpiration efficiency. So for me and for my understanding, what I anticipated from the bits and pieces of the evidence which we collected, transpiration efficiency really to me looks like is the continuous interplay between the mass accumulation processes and photosynthetic available or sink available for the photosynthetic and with the interplay with the tissue expansive processes, which where the hydraulic processes has the rule. So whatever it is, I believe that this is the front line of research across the globe now. And what is sure is that we need appropriate tools and methods to study these phenomena further. So that brings us to the relevant tools and approaches which we have developed at ICRISAT. So if we hypothesize that the transpiration efficiency is the function of the water transport and ability to grow in environment, then these mechanisms we can study at different level of the plant organization. We have a LYSI-scan facility, which some of you will visit this afternoon, some of you have visited, where we can really in a high throughput and very detailed manner investigate what is happening, how the canopy expansion is happening in time, and what is the water demand to support these expansive processes. And then we have the lysimetric facility where these phenomena can be investigated in more integrative manner. So this LYSI-scan has proven very useful when we wanted to follow the development of canopy in time, what is shown on this graph here, on the example of parallel millet ecotypes, which clearly differentiate in their way how they develop the canopy in time. And that well coincides with the origin of agroecological zones. Where do they come from? So this is really positive news. We can, based on these techniques, we can differentiate between different agroecological plant types. But there is still a component which we don't understand well, and we need to really take care of this. Because if we screen the same material during the summer where the VPD of growth is really high, then we cannot really differentiate between these ecotypes in the same way. So this system is really very useful when we want to look at the genetic basis of these traits. What is shown on this picture here? So for the case of chickpea, we were able to identify the major QTL on LG4, which really explained the majority of proportion of variation for canopy growth in time. And this was really curious case because the same hotspot region on LG4 has been previously shown to influence the root density and chickpea yields in the stress condition, water stress condition. This doesn't work. Okay, so then another equipment which we have at ICRISAT, which you will see, is the huge set of the low cells where we can gravimetrically assess the plant transpiration responsiveness to VPD in a real time and importantly outdoors. So before we had this, This is not fair play. I'm losing time because of not working things. So before we had this facility, we were destined to measure the plant transpiration responses in a very artificial conditions of the growth chamber. Of course, we were able to differentiate between different genotypes. So we only hypothesized that if the plants behave like this in the growth chamber, if they can limit their transpiration in growth chamber. Okay. So we hypothesize that these plants has the capacity to restrict their transpiration during hot parts of the days as well. But this outdoor system was really the first real world system which allowed us to confirm these differences between the genotypes. You see these small differences, but not only to confirm these differences, but also quantify them. So again, when we were thinking about what could be the genetic basis for these differences dealing with the transpiration, uh, we were interested about whether they are determined by the drought tolerance QTL in ferromillet case, which have been identified long before, and we have a set of near isogenic lines which was segregating just in the region of LG2 to investigate here. So when we investigated this genetic basis of these TE components dealing with transpiration, indeed, many of them were mapped into this drought tolerance QTL. But the biggest surprise was this huge peak of highly significant QTL, which basically explained a huge proportion of the canopy structure. So this is something what we didn't have the access before. And that opens a big question: what is the role of canopy structure in the processes determining transpiration efficiency? Oh my goodness. So where should I do it? Yeah, so another part of our very exciting research is the interspecies comparison. In this case, we were interested about how maize compares to our mandate crops, sorghum and farro millet. So this was again very interesting case because what we found is that this is really not fair play. Can you move it, please? Okay, because what we found out is that maize across a different water limiting environment attained significantly higher transpiration efficiency from all these crops. So our question here was, is this really the result of the 100 years of intensive selection in the maize crop? And if so, is there any simple way, relatively simpler way in which we can bring our mandate crops to the same level, but without requiring 100 years of the selection? And if yes, what are those components? So at the end, the most important thing, how do we apply this research then that we can see the impact of what we do in the fields of our stakeholders? So this is dealing with much more complexity and diversity than what I was showing till now. If we are serious about doing research for impact, we need to be really very clear about, okay, let it be like this. We need to be really very clear about addressing the main demand for our crop products within the environment or within the context of given society and given market situation. We are dealing with the production of these crops in an environment where each of these environments have very likely different production constraints. Then only we can start thinking about how to address these issues with the breeding methodology. And what I was talking till now is really only very small part of the process of accelerated selections, which matters for addressing these two first components. And finally, of course, the farmers have to choose our product. So if we do something wrong in this issue, in this product delivery pipeline, then we are stuffed anyways if our products are not accepted. What I really mean to say here is the transpiration efficiency alone or any other trade alone will not feed the world, and we need to be really very clear about it. So if we go further, what could be the possible tools to tell us whether what we do really makes sense and whether it has the viable probability that it can be applied in the crop improvement program. So this I want to demonstrate for you at the similar slide or same slide which Peter Carver was showing you yesterday. So if we want to evaluate the putative effect of the system intervention, in this case, we want to see what happens if we grow the genotype which develops a smaller canopy. We can actually translate this information in the appropriate functions and link it with the information of management and environment in the context of our target population of environment. Then we can run this information through the model and investigate and quantify what is the benefit of growing a crop with shorter canopy. So this is what I have shown on this graph here. On the x-axis, you have the original yield of the genotype which develops larger canopy. And on the y-axis, you have the grain yield change due to a growing plant with smaller canopy. So here you see very clearly that in the low production environment, having plants which develop shorter canopy or smaller canopy very likely translate into the yield benefit. But we need to be very careful because our mandate crops are usually grown for dual purpose. So we need to do the same exercise for stover yield. And then we need to be very careful about the evaluation of the possible trade-offs. And finally, analyze what is the economic viability of this intervention and most importantly, where. So this slide gives you the notion that we actually need very different plant mechanisms in very different agroecologies to address the differential demand. So this I want to demonstrate you and quantify for you on this slide. So on the case of sorghum, so sorghum in India is grown for dual purpose, for stover to feed the cattle and grains to feed the people. And there is differential demand for these crop commodities across India, what is shown on this graph here. So in a greenish areas, there is more cattle, so people grow sorghum more likely to feed the livestock, while in those reddish areas, people grow sorghum for their own consumption. So if we have tools and we have know-how how to develop the plants which maximize the grain production with a trade-off on stover production, and we can do the opposite. We can maximize the stover production with allowing certain trade-off at the level of the grain. So what is the benefit of having two production pipelines instead of having only one product? In this case, our social economists help us to evaluate that doing these two differential products to address different needs of our stakeholders directly translates into the 12% higher income for people living in this area. In this case, it's worth of $200 million of income for these people living in this area. So in a summary of these third and fourth points, I was talking about the fit-for-purpose technologies, which now open a new dimension of research, which was not even possible before. I was talking about, for example, the curious case of the rule of canopy structure. I was talking about the product development pipeline, and I sincerely believe that there is really no major silver bullet left in agriculture Rather, I believe in highly focused and strategic steps to part by part enhance the demands of our stakeholders. And I was talking about the modeling tool, which help us to assess the lucrativity of and impact of the system, any system intervention, be it the intervention in management, or be it the intervention in a crop genotype, developing new crop types. So I hope that at the end of my talk, you are convinced that at ICRISAT we have developed the necessary understanding at the scales which is highly relevant to support reasonable quality of the research, and still at the scale which is relevant to assess the crop improvement programs. So with this, I want to thank you. And if you want to know more about our team and about the people who all contributed to this presentation, please visit us at our website. And with this, I thank you.
Thanks, Jana. Don't disappear just yet. Jana, just wait a minute. Actually, Jana's finished with a few minutes to spare. So if there are a couple of quick questions, we can take them now. So if anybody has a question of clarification or a question for Jana, we've got a couple of minutes. Please go to the microphone. Otherwise, we'll have a general discussion later. Is there any questions immediately? One at the back. While he's coming up, I did like the use of the term lucrativity.
Okay, I am P.S. Zaidi from CIMIT, and Jana, congratulations for this excellent work, and especially the comparison of different C4 crops in terms of transpiration efficiency. And maize is, leading in all different soil moisture regimes, which is, fair enough, because maize, particularly the single cross hybrids, high yielding hybrid, they are tailored in that way. But the another side of the coin is that when it comes to stress-prone environment, that's where this sorghum pulse millet take lead. So, with your experience, what is the trade-off in terms of transpiration efficiency?
Oh, this is still open question. One thing what we are really planning to do now with the collaboration with Pioneer guys is to parameterize these crops into the crop model and run it across our locations of interest and really see where is the higher chance that the maize will be more beneficial for our stakeholders, and where is the higher probability where other crops will bring more benefits to the farmers. So we do not really compete in the same agroecologies, but rather strategically align our efforts in a benefit of the farming community. So we don't push sorghum and millet in agroecologies where we know that the maize can bring farmers more benefits, and vice versa, that we push our crops with the best focused targeted plant types to the regions where really people cannot benefit from the maize any longer. But it's a really good question, and we will have to discuss this maybe further.
Thanks, Jana.